Okay. Yeah. Okay. All right. Well, good afternoon, everyone. This is on? Yeah. Good afternoon, everyone. Thank you so much for being here. My name is Marilyn Ahun, and I'm a professor here in CORE in the CHHD program. And it is my pleasure today to present Dr. Fackson Mwale, who will be talking to us about the role of LINC-N in osteoarthritis and degenerative disc disease. So Dr. Mwale is a tenured professor and the graduate program director in surgical and interventional sciences in the Department of Surgery here at McGill. He serves as co-director for the Surgical Innovation Concentration and as a senior investigator at the Lady Davis Institute. His long list of accomplishments include serving as the past president of the Canadian Orthopedic Research Society and the Canadian Connective Tissue Society and co-founding Trepso Therapeutics and Interventional. Dr. Mwale has made significant contributions to the fields of orthopedics and regenerative medicine, with a particular focus on intervertebral disc degeneration and osteoarthritis. His talk today will explore the therapeutic potential of LINC-N, which is a novel peptide that targets intervertebral disc degeneration and osteoarthritis. So thank you very much, Dr. Mwale.
So thank you very much for that wonderful introduction. Okay, so these are my disclosures. So I'll start talking first about back pain, which is ubiquitous. If you don't have it now, you will have it at some point. So yeah, so it affects a lot of people, especially with the aging population, and it's very, very costly. And these are just the numbers from the US and Canada. And one of the things I'll be talking about is the intervertebral disc, and it's the one that's associated with back pain. So, so this is a human intervertebral disc, and this is basically just I just wanted to impress upon you just how huge it is. And this is on the lumbar, and you can see that here in the center it's a nucleus pulposus, and normally this is where it is quite gelatinous. And around this are the annulus fibers. And then you have the end plate. So it's quite huge. You can see that in terms of diameter, it can be about 4 centimeters and about 10 millimeters, 7 to 10 millimeters thick. So it's quite huge. And one of the things, what's happening in the intervertebral disc, so there are cells in there that are called disc cells in the nucleus pulposa here in the center. You have got these cells that are nucleus pulposa cells. So basically what they are doing is that throughout life, they are making matrix. And mostly you have got aggrecan and you've got type 2 collagen. But they are also making proteases. They are cleaving these molecules. But the rate at which these molecules are made it normally equals the rate at which they are being degraded. And so, but sometimes, as we saw as in this degeneration, what happens, there's more degradation and it tilts the balance. So when that happens, you can see here's a normal disc, really nice, and the disc height is very nice there. But when there is degeneration, you see that there's a decrease in disc height. So when I talk about the animal models, one of the things I'll be talking about is this decrease in disc height. So if you see % decrease in disc height, this is what I'm referring to. Because the idea is to try to find a way to bring this disc height up. Because if it comes down the way it is here, there's all sorts of structures around that can be affected and that affect back pain. So there are a lot of causes of this degeneration. So it can be cellular. We're born with notochordal cells, for example. And then by the age, a certain age, we start losing these notochordal cells. And some people say that that's one of the reasons you start this degeneration. It can be nutritional, it can be mechanical, or it can be genetic. Yeah.
Degradation and
Yeah.
and production.
Yes.
And it's the ratio between the two. Why is there degradation from the beginning? In a bone there, in the bones, there isn't.
Well, yeah. Well, in the disc, it's basically just turnover.
Oh, I see.
Yeah, it's just turnover. Yeah. Yeah. So these are a lot of the causes of the current treatments for intervertebral disc disease when there's pain. There are a lot of current treatments, as you can see here. You've got most of them are simply to relieve the symptoms because when patients go to the hospital, it's the pain they go there. They don't go there because they have got a decrease in the intervertebral disc height. So you can see these are so if these don't work, there's also surgery. But what happens is that if you switch, mostly there's a lot of fusion of the spine. So what happens is that this can result in altered biomechanics, and then the degeneration continues. Because when you fuse the spine, you alter, you basically change the biomechanics of the spine. So there is no drug that has been approved to address underlying disease. What about repair? What are we doing in the disc? What's the approach in the lab? So there are different grades. So you see there, grade 1 is a normal disc, and in the center you see the nucleus pulposa is full of water, and that's how a normal disc should be. And it will resist compression because it imbibes that water. And then you see grade 2, you start losing that gelatinous, the water, and then grade 3, you start having all those fissures around. And then if you get grade 4, you start seeing all sorts of hell is happening in there. And grade 5, you pretty much can't try to repair that biologically. Yes. So these are actually actual human, yes. No, this is just a yeah, post-mortem. Yeah, they died. Oh, they died. Yes, yes, yes. Yes, yes. Yeah. Yes. And so then if you are trying to address grade 2 or grade 3, you can probably what you need is a growth factor to stimulate matrix production and some sort of gene therapy. Maybe that's the approach you take. But if you go to grade 3 and grade 4, you have fissures in there, and you need to put in something, not just stimulate the cells. You can put in a support scaffold to help with the mechanics. But for grade 5, you can't repair this biologically. You just have to remove it. As you can see, there's a lot of problems with the grade 5. Now, a couple of years ago, we came across a peptide. So what you are seeing, these are called proteoglycan aggregates. So that is called link protein, and link protein stabilizes the interaction of this binding of G1 domain to the whole structure. Now, what happens is that during tissue turnover, there is cleavage of these 16 amino acid peptide. So a couple of years ago, a group in England took this and then they gave it to some cells chondrocytes, and they found that there was stimulation of proteoglycan. So we continued with this peptide and trying to figure out if this can be able to work in the intervertebral disc, in the spine. The whole idea is to try to see if we can stimulate matrix production with this peptide. And one of the advantages is that it is cheap to make. It is a peptide. It's not a recombinant protein, and can be able to see if this. So anyway, so there are a whole bunch of properties that have been found of this peptide. Just to summarize a few here, it is mainly released by cleavage by a protease such as MMP cleavage. It's produced both in articular and intervertebral disc. That's why when I talk about both osteoarthritis and intervertebral disc, because it's found in both. And it seems that the idea, nature's idea, was that this was some sort of messenger to say there's so much matrix product degradation, and once it's cleaved, it will go and tell the cell to make more matrix. So at least that's what we think. So the questions we were asking at the beginning was that if we supplemented this peptide, would this be some sort of therapy for both intervertebral disc degeneration and also osteoarthritis? So the first thing we did a lot of in vitro studies, but as you know, you can do all the in vitro studies you want. You still have to go in vivo. So we went for a rabbit model, and we did that model in Chicago, and what with Dr. Koichi Matsuda. And so what you see here is that we have a control group, and we puncture the spine. These are rabbits. We puncture the spine, and then after 2 weeks, we inject with either saline or this concentration of Lincoln. So we inject to the degenerative. And then bi-weekly, we do X-rays. Yeah, that's a great question. So we did it by annular puncture. So we simply puncture the disc, and when you puncture the disc, you start getting degeneration. Yeah.
That fast.
That fast. Yeah. 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 So one of the reasons, of course, the rabbits are we're trying to look for a bigger animal, and it has got similar. The discs of the rabbits have some similarities to the human disc, and a lot of people have been. Of course, an ideal situation would be something that is not a quadruped. That's one of the limitations. But in terms of biped, we would have to go. There are very few animals we can work with. But the rabbit is a compromise for the.
And you're sure that just puncturing, what happens afterwards is exactly the stages of degeneration. It replicates the stages of degeneration that you have in the human, the 4 or 5 stages.
Yeah, it's quite very similar, very similar to the human condition in terms of injury. Because once you puncture, what happens is that this is an injury model, basically. And what happens is that you get all the usual suspects, the cytokine IO1, TNF alpha, they go up and they start to degrade the disc. Yeah. So you can see that the first thing that we do, so you puncture there and then you inject there after 2 weeks, and you can see that this is the saline, and you see that there with Lincoln there is an increase in this height, which was quite very interesting to see. We are seeing all these nice things in vitro, but we had never seen it in vivo. So there's this increase in this height. And when we looked at what sort of molecules, we took the tissues we analyze them for what's going on to the matrix molecules, to aggrecan, and also to type 2 collagen in the nucleus pulposa and annular fibrosis. So all I'm showing you here is aggrecan messenger RNA. We have done also a protein, but you see that there is stimulation of both in the annulus and nucleus pulposa, which is a good thing. So that's what we're looking. And then when we looked at the proteases, because as I said at the beginning, during turnover, there is a proteolytic cleavage as well. But during this degeneration, there's an increase in these matrix MMP proteases. So you want to bring them down. And you can see how MMP and ADAM-TS4, which are the usual enzymes that go up, they both come down with linkage in the annular fibrosis as well as in the nucleus pulposa. So while we are working on this, we didn't know that there was another group in Atlanta that was also working on it. And so they found similar things. And one thing that they found, and this they use rabbit cells from rabbit. And they found that actually Lincoln is actually dose and time dependent. And they published in JBC, which was reading a very nice paper. Now, we're you can do all the animal stuff, but eventually we are trying to deal with humans here. So what about humans? So we took the human disc and we injected, so the human disc, so these are already degenerated, so you don't have to puncture anything. So you get from, so first we took the human cells, and in this case, we are looking at protoglycan synthesis. And what we find is that these are cells from nucleus pulposa or annulus fibrosis. And what we find is we can stimulate cells from humans, and we're trying to basically find out what's the optimal concentration. And we found that the optimal concentration was about 1 microgram per ml. And so going ahead, you find that I'm using this kind of concentration. So that's the rationale for it. And the other thing we did was that, okay, well, what about donors? We look at different donors. But one thing we found is that there is protoglycan synthesis, and this is the whole human dish. And with that, there is stimulation of protoglycan synthesis, but there is variation. You can see this donor, the stimulation not much, but this donor is very responsive, and there's that donor. So it's something that we have to bear in mind. Yes. So we go to harvest from cadaver, and then we analyze those views. Yeah. No. The idea is to, if this works, we're just trying to see if the human disc can respond, and if they can respond, then for those people who have problems with their spine or their disc, which is degenerated, we simply can inject with Lincoln. So that's the idea. But we have to go to the human disc. When somebody dies, they are still alive for quite some time. And we can harvest the cells and we can get the human disc. So this is a program that we've been doing for quite some time. So the other thing we wanted to find out is that how long does Lincoln stay in the human disc once you inject it? And the way we did it is we attached a fluorophore so that we can be able to see if Lincoln is there. And then we changed the media for 5 days, and then come back and check. We detect by fluorescence. And when we took different punches, and you can see that there is retention in different punches. You can see that 1, 2, 3, depending on the human, and you find that actually there is retention. That's the central punch, and the retention according to the different locations. So this is important because if you're going to have injected, you want to make sure that it stays in order to do its job. The other thing we wanted to know is that what about diffusion? How does Lincoln diffuse? Because if you inject it and it goes out immediately, then you won't have much of an effect. So we did diffusion again. You can see the human disc. So we were thankful with the Association with Transplant Quebec, and that is a pre-injection before we inject Lincoln, and we injected Lincoln, and so this color is just a color that we attach as a fluorophore to Lincoln, and you can see that here. You see that it does diffuse also around the disc, and it's actually stopped by the inner annulus. And so it does stay in the disc, and this is important in terms of drug development and to see if this is quite something viable that we can develop therapeutic. The other thing we wanted to find out was how long does it stay? Can it stay in the disc? And we looked at, so one of the things that was easier in terms of following proteoglycan is that we can label with sulfate as a surrogate for proteoglycan synthesis. And you see that it can stay, and we found that it can actually stay for about 9 days. And we stopped here, and so and this is compared to the control, and this is different levels from the spine. So when you you inject with Lincoln, you do get it to stay inside, and you get it to diffuse as well, so that you understand where it's going. Now, what about the? We were asking ourselves, so is this is Lincoln, is it specific? So what we did is that we decided to reverse the sequence. We decided to scramble the sequence, and we found that actually it is sequence specific. When we scrambled or reversed, we lost the activity. So, and this is with human cells. You can see the synthesis of proteoglycan, and that's Lincoln. You do anything to it, you lose the activity. And of course, we are quite very curious. So this is work that was done by Dr. Rahul Gowri, who is now a professor at McGill. And we sent him, he was my PhD student, and we sent him to Sweden to find out how stable this molecule Lincoln. And one of the reasons that they have got all these $1 million machines that we can follow Lincoln. So so he looked at, so he Dr. Gowri, when he came back, he said, Fackson, I want to talk to you. I said, and he told me that Lincoln is not stable. I said, thank you for telling me on Friday. So he found that if you take Lincoln and you put it, it is stable when you put it, expose it to nucleus pulposa cells. But if you expose it to annulus fibrosus cells, you lose the activity. You see, immediately you have lost the activity, and we said, what's going on? We are seeing the effect, but it's not stable. And immediately we try to find out what's going on. It doesn't make much sense. So we found that there was a small a peptide of this mass, 1922 Dalton, which was accumulating, and that there was fragmentation. So with a peptide of this mass, we thought, okay, what's accumulating? Maybe this is the possible first sequence, and it's 8, or it is a second sequence. And so we said, okay, it is being cleaved by annulus fibrosis cells. And then we found that if we are basically getting this 8 amino acid peptide, so it's coming from 16 to 8. And so we have what was being generated. We took all the enzymes, the usual a lot of enzymes, and see which one is cleaving link N. And we found that it was only cathepsin K which was cleaving link N. And then we did all sorts of it was kind of Sherlock Holmes, which was exciting for a scientist. But eventually we found that actually the active part is the first 8 amino acids, and the cleavage is happening by releasing that 8 amino acid. So when we get that 8 amino, of course we wanted to find, is this active? And we found that indeed that was active. And since we are very original, we called it short linkage. And so that's where the short linkage came from. So the first 8 amino acid is where the activity is. And the rest, the 9 to 16, we find that it doesn't have the activity. So we sighed some relief, knowing that we had solved the puzzle. But of course, we had to go back to square one to find out, okay, does it work? So we took these bovine tails, this from bovine tails, and we treat them with trypsin in order to degenerate them. And then we inject them with short lincoln. And then after 5 weeks, we analyze to see if that is working. And so what we saw is that, as you can see here, you can see that that once you use the short lincoln and you compare that it does have activity, and that you can compare with the long lincoln, and that's where the activity is. So now we had to go back to the rabbit to see if it's working in the rabbit. And, you know, it's again, another $100,000 to do this. We go back to the rabbit, and we go back to Chicago, and then we do this experiment with Dr. Koichi Masuda. And we found that, so this is the design of the experiment. You can see short lincoln again does stimulate this type. And yeah, so when we look at the protoglycan content, you can see that short lincoln also it doesn't matter if it's in the nucleus pulposa or in the annulus fibrosus, you get stimulation of protoglycan content. Now, again, we looked at metrics. When you look at metrics here, short lincoln, you can see the stimulation here of metrics that compared to the non-function. So basically, the lincoln works, and that's where the biological activity lies. Now, I'm going to move to osteoarthritis. And we said, okay, let's move and see what is happening with osteoarthritis. So osteoarthritis, you can see there are a lot of causes. And again, similar to intervertebral disc degeneration, most of it, the current treatment, all those lifestyle modifications, you're both symptomatic, basically trying to get rid of the pain. And the surgery, all sorts of surgery that works, but there is no disease modifying that exists. So what we did was we took cells. So this is Dr. Michael Grant, who is the research associate in my lab, and he took chondrocytes which are found in the articular cartilage, and then he stimulated them with scans, and you could see, and this is from osteoarthritic patients, and he stimulated and you can see there an increase in proteoglycan, and it actually depended on the dose, which was quite very nice to see that it's working in the human. So again, this whole thing is basically saying that we then took explants from cartilage and analyzed them ex vivo. It's one thing to take cells and stimulate them, but I think it's nice to be able to see in their native environment how this is working. So we took these flags and we mimicked degeneration by using I1 beta. And interleukin-1 beta is involved in osteoarthritis. It's one of the major players which destroys the cartilage and is involved in the whole destruction of the disc. So as you can see there, with I1 beta, you see a decrease in proteoglycan synthesis. When you use lincoln, you add lincoln, you see that there is an increase in the synthesis of proteoglycan. Again, we have the rabbit, the poor rabbit, but this one is an osteoarthritic model, and this time we went with Dr. Sheila Laverty in St. Catharines, and we did this experiment in which you have the rabbit and you do an anterior cruciate ligament transection, so that will mimic degeneration of the joint. And then after you do anterior cruciate ligament, then we add we inject with short lincoln every three weeks. And one of the reasons, you remember that in the disc we just injected one, but one of the reasons we're injecting here 3 times is that in the joint has got these lymphatics that drain things. So if you put in peptide, it will be drained. So we constantly had to go and be injecting. And then we look at after 12 weeks. So after 12 weeks, here is the gross morphology changes of the articular cartilage. And what you are seeing is the sham. Those are the condyles. And you're seeing the condyles there. And you can see that for the selling, you can see the selling here, how degenerated. So this is just a, and if you compare with short lincoln, so short lincoln came in different flavors, 2 major flavors. One was it's still degenerate, but it's quite good. It's almost close to the sham. And then you have another one which is degeneration. But you can also quantify this, and the quantification is such that for normal, you put it at grade 1 because you are using something called India ink. And for fibrillation, you get grade 2. And when you get erosion, like this is erosion, you start getting that kind of, or those kind of grades. Now, if you quantify that, so we had to do all this blinded, and we sent this to, this was done by Professor Mwale. Some of you know him. He's one of our best professors in statistics, and he works with company to do it. So when we analyze this statistics, we say, well, okay, what's going on? And what you see, so these are different compartments. You can see the compartments there. And when you look at the different compartments, what is going on is that you can see in terms of color, you see fibrillation and you see erosion. But what was interesting, immediately apparent, was that if you use short lincoln, you don't see fibrillation. You don't see fibrillation. You can see it doesn't matter for which compartment. And so this was quite interesting. You see, in the selling, except for the medial tibial plateau, this compartment, in the rest, you see fibrillation, but with Lincoln, you don't see fibrillation, which was really interesting. And you can go to histology. You can look at histology. And if you look at histology, that is a sham. You see the short Lincoln again coming in two main flavors. You've got this one and you've got that one. But most of the selling were around this flavor. So you can quantify all that. And what we saw is that actually short Lincoln prevents protoglycan loss in vivo. So as we are doing all these experiments and we are going to conferences and people are getting excited, and every time everyone would say, what are the mechanisms? Tell us, how is this happening? They're rare, phenomenal, good, but how is it happening? So we are lucky to get a grant, another grant from CHR and to study the mechanisms. And we were, working hard to get those mechanisms, and my students were getting all excited. And when we were about to publish our work, the paper came out, which had done the mechanism. And this was a paper again by the group in Atlanta, Dr. Tim Yoon's group. And I remember one of my technicians just came with the paper and said, but that's research. So, and they did an elegant way, really beautiful way. So they were calling it a link peptide. And so basically, they found that what is happening is that when you bind, it binds to BMP receptor 2, and when it binds to BMP receptor 2, that it stimulates endogenous BMP 4 and 7. And you can shut down this system down at any point, depending on the inhibitor. And that BMP 4 and 7 will come out of the cell, and it will go and bind to BMP receptor 1. And when it binds to BMP receptor 1, and then it stimulates the SMAD1/5 and SMAD1/5. And so that's how it's making all those matrix molecules, collagen and aggregate. And it's really beautiful work. They got awards for it. And you can they showed that you can shut down by noggin, which binds to BMP. You can shut down with noggin. You can shut down with SMAD, and you can shut down with SIM-MRA. So really elegant way. We did that when we found very similar, except we used the human cells, and we find very similar. So, and you can see this is the work that we did, and we found that the short link can actually also that it induces BMP2 and BMP4, and you can see there the actual. So this is a cell autonomous loop. Now, the other thing we were wondering is that so this was done by Dr. Michael Grant. One of the main players is I1, interleukin 1. And so I'm cutting a very long story short here. So we found that actually Lincoln interacts with interleukin 1 beta. And we did a whole structure, that's the crystal structure of I1, and those are the bindings, and that those are the main residues that are involved in the activity. And we can see, we also did immunoprecipitation. We could find that the I1 is binding, is actually binding to Lincoln. So which was quite very interesting. Now, with all this talk about BMPs, which are bone morphogenetic proteins, when somebody hears about BMPs, you think about bone formation. So the question that immediately people were asking is that, are you not forming bone? if you're getting that BMP, Lincoln is making the BMP, are you not forming bone? And if you're forming bone, it's not good for cartilage, and it's not good for for intervertebral disc. Again, the wonderful group in Atlanta showed in a wonderful paper, they showed that actually with Lincoln, that it does not stimulate osteogenesis, you can use BMP2, you compare it with Lincoln and BMP7, and you look at osteocalcin, and you look at alkaline phosphatase activity, and you find that actually this is Lincoln, very, very minimal stimulation of those. So that's very interesting. It seems that the cell autonomous loop of BMP4 and BMP7 is not leading to osteogenesis. Now, this is the last part about pain. So what about pain? So I'm just giving you here of the intervertebral disc. In a normal disc, you have very little it's a neuro, and you have the blood vessels. They're just at the periphery of the annular fibrosis. But once you start getting degeneration, what happens is that you you get hyperinnervation, and it's that hyperinnervation that and that is mostly because you start stimulating all these cytokines that start going up, and so also it feeds into the whole pain loop because it has to go. It goes through the dorsal ganglion, dorsal horn, and back to the spine, to the brain, and then comes back. So that is pretty much just an idea. So the important thing is you have in the disc, you have hyperinnervation. And this, the main player is I1, interleukin-1 beta. And so when, so we looked at the, again, we looked at the human intervertebral disc, and we found that with the different grades of degeneration, you get, as you can see there, you get an increase in NGF with grade of degeneration. And you can see NGF here. And when we did we go back to stain the samples from the rabbit, and we find that here's NGF is going up. Once you do the function, NGF is going up, and these are related to pain. But this, interestingly, is part of the inflammasome, and so that opened a whole new chapter for us to see how the mechanism is working in terms of the inflammasome. And so this is again by Dr. Michael Grant, and he found that, to cut a long story short, is that you get stimulation of NGF, you can shut it down with Lincoln, and another player in pain is BDNF, and you see also TNF, alpha, and interleukin-1. So you can shut them, and so this is just in vitro study. And one thing you can do is you can induce NF-kappa B. So this is involved in pain, and you can stimulate that by adding interleukin-1 beta. And we can see that in vivo, on the analyzing with the cells in vitro, you can stimulate, you can activate NF-kappa B, and with different doses of Lincoln, you see there's a decrease in NF-kappa B. And as we were doing that, so this was cultured human chondrocytes. And as we were doing that, having to remember that, okay, there is a binding with I1. So we start thinking that, okay, how is this working? So it opens the whole point of the inflammasome, which is a very exciting area. So this is the inflammasome here, and it's involved in activating I1 beta, which is inactive, to make it more active. And when it comes out, it's the one that makes all those problems. So, so that cascade can actually be activated by dumps, and it goes through the toll-like receptor there, and it's helped by its partner CD14. And you need those two to recognize what you have up there, the dumps, etc. And those lead to the stimulation of NF-kappa B. So that's how it is working in terms of the NF-kappa B. I mentioned NF-kappa B because it is involved in inflammation, I1 stimulation. So this mechanism is quite very important in terms of our understanding. So here we found that short-linked actor inhibit. You can stimulate that whole thing by using LPS, lipopolysaccharide. You can induce NF-kappa B and activate that, and you can shut it down. But with that whole cascade, you see that there is a stimulation of caspase 1, and that you can also see I1 beta being stimulated. So this is all in the inflammasome. So it all gets to see that there is also an effect of linkage in the whole inflammasome, which is this multi-component, which is involved in inflammation, in pain, etc. So we found that was exciting. So what we did is that, okay, we had another professor in Chicago, Dr. Im Sampen, who is an expert in pain, and so he was running some models of pain. And so we said, okay, what happens if we inject Lincoln, generate in this pain model, and so this is called a von Frey basically. And what happens if can you shut down the whole pain thing? And you can see here that's the sham, and that is with Lincoln, the red one. And normally the sham, if you use about that amount of grams, basically what happens is that the mice will withdraw according to the load that you apply to them. So if they have that load of about 4.5 grams before they withdraw their pause, it's much better than having, you can see the PBS, it's quite very low. And this is called allodynia. And what we find is that there was a rapid reversal of mechanical allodynia by single Lincoln injection. So, and of course, we were getting all excited to go about mechanism. What is happening inside in order for all these things to happen? So what we did is we first isolated murine DRG neurons. And so on top, I'm just showing how we isolated them. And then what we do is that we challenge them with the I1 beta, and you can see that is a real live streaming of what is happening to those neurons. And you can see with I1, you get the calcium influx there, and which means there's a lot of hyperactivity. And you can see the control there. And with Lincoln and I1 beta, you get a decrease in that activity. So that was very, very interesting to see that actually it is happening with the inflammasome, and it's also happening in terms of we can do another kind of experiment to see that it is also being affected. So this is just a summary in terms of the inflammasome part and in the intervertebral disc where we did, and it seems to be shutting down when you use with Lincoln. The NF-kappa B is suppressing the NF-kappa B and caspase 1, and it's also shutting down I1 beta as it comes. So this is important because this is what will lead to all the havoc fully to pain. So I'm just about to finish. So one of the things that we do is that to try to compare with other well, that are being used with the same type of paradigm. They are anti-inflammatory, and you just pick what to compare to short linkage. And there's also use of steroids. There's also biologics. And one of the advantages of is that it's quite very cheap to produce. And you see those kind of comparison of an ideal characteristic for a therapy. So this is just what I was saying, and it's basically related to mechanism of and pain pathology, and that the efficacy in repair and inflammation, we are able to show that in vivo, and the mechanisms of linking and cellular and what sort of insight, and we have delved into the mechanisms, how it works, and we have also looked at the preclinical evidence in vivo and sort of in vivo studies. So the conclusion to all that is that Lincoln offers a multifaceted solution to interventional disease and OA by promoting this tissue regeneration, reducing inflammation, and alleviating pain. So a lot of people have been involved, and one of the pioneers who helped us with Lincoln, Professor Peter Raffley, who passed away three years ago. So he was one of our main mentor to help with all this. And this is Raul, who is now a professor at McGill, and we collaborated with Professor Lisbeth in order to find a short Lincoln. Without going to Sweden, we may not have done that route. And yeah, so again, a lot of people are involved. Dr. John Antonio, he's an orthopedic surgeon who I call my twin, and help with the Koich Masuda, a lot of funding from PIHR and NAS and all the, so really much appreciate. So thank you very much.
Thanks very much. That was interesting and a lot of work for a very important problem. So it sounds like your research program operates in a space where you can do a lot of science sort of in vitro, in vivo animal models, and you even have this unique, relatively unique situation where you can have human tissue, which not everyone can explore. And so at least in the degenerative disc work that you showed, you really brought it all the way up to, it seems to work in these sort of human tissues ex vivo.
Yes.
It seemed to me that the next step would be seeing in vivo humans, like actually doing trials of some sort to evaluate whether it truly impacts, pain or important outcomes like that. Where did you take that? Like, did you go to industry to do that, or you just stop there and hand it off to someone else to run with it?
Yeah,
because it seems very promising, sort of experimentally, but then trying to bring it into the clinical efficacy realm seems like the next.
That's a wonderful question, and so that's why we started this company, Therapeutics. That's what we are doing at Therapeutics, basically.
Okay.
Yeah.
So do you have trials ongoing, or?
So that they are, because we are targeting 2 indications, and so we have to do some additional work to be able to go for the 2 indications. So at the moment, and we have help from the government to try to see how we can get in the clinic. It's a lot of work, sure. And but at the same time, it requires, once you go into clinic, it requires money.
It gets more expensive for sure. I don't think
that's where we are in terms to try to raise funds, and it's a lot of money to start the first part.
Is the print just? I'm naive about this space. Is just as a therapeutic approach, injecting something into that space. Is that like? Is there precedent for doing that in living humans, or is even that delivery approach something totally new?
Yeah, that's again a very wonderful question. So it's it there are some studies that are being done in terms of trying to inject, but that's one of the limitations because it's not you know, you don't have a lot of biologics that are being injected in the spine. So you have to interact with the FDA and try to. But one of the things that the NIH and the US government has put in a lot of money because they want to solve this, and also to try to see, because of course in this you don't want, no one wants to come to the hospital injected every so often. And if we are trying to find out what job we're doing at the moment is to do a control release. And so before we can jump into humans, we have to validate the control release and what sort of concentrations we need. So it takes a long time. It's nice to do in the rabbit. We know the sort of concentration that we use, but are they going to translate to the human condition? So we are working with control release so that at least if you are going to inject, you are going to inject maybe once, and there's a control release in place, especially in the joint. Osteoarthritis is a bit more difficult for that because there's a lot of clearance. And so we are developing this nano technology to bind. But whenever you you develop that kind of nano, they send you back to go and check if it's happening, it's working in rabbits and how it's working. So that's one of the things that we are doing. And then we can go into human. Because in human, we can't just inject a naked peptide. We have to inject in a sort of control release.
The questions. Thank you very much, Dr. Mwale. This really sounds like an interesting topic indeed. I'm not a basic scientist myself, so but you still managed to keep my attention to the end. I just had a couple of questions. One of our colleagues here was asking how you, sort of created the degeneration in the rabbits.
Yeah.
And you said it was an injury model. And I was just curious, is that how it happens in human beings as well? How does this degeneration begin? Is the injury always a starting point?
Not really. It's not, well, it does happen, injury does happen, but like I showed before, in human, in terms of spine, there are a lot of factors, a lot of things that can lead, but it's also very slow in humans. And of course, aging is one of them, one of the major ones. And but for osteoarthritis, we have post-traumatic OA. So you have some sort of trauma to the and it starts to degenerate. But a lot of it's a lot of multifactorial. It's just that in the it would take a long time and a lot of money to keep rabbits aging. so the injury is a surrogate, but still, if you injure the spine, yeah.
Okay. In fact, I thought you may have listed it at the beginning of a few minutes later. And I have a second question.
Yeah.
And that is, I feel that, just anecdotally, people who have these kinds of problems, they often these days receive an injection for treatment.
Right.
And what is in that injection if it is not a linked end yet?
Yes, it is. Yes. So there's also injections of cortisol, and
okay, cortisol.
Yeah, cortisol, and there's Simvix that is injected. So those are mostly to stop the pain.
Okay.
Yeah,
I
they are
mean, I know
directed.
cortisol is in the context of arthritis.
Yes,
but
context
that
of
is it. That
arthritis.
is-
Yes,
that's all. Okay.
yes, yes, yeah. So what? Just going. Um, so just going back to the question about the injury. So this is more like disc herniation. Really it's quite similar to disc herniation. So we could say that this is a disc herniation model in the spine when we injure the spine. Because when you injure the spine, what happens is that the nucleus pulposus they come out, and you have a certain amount that you take out. So it's a kind of disc herniation. And whenever there's disc herniation, what happens? In most cases, you start getting this degeneration. Yeah.
Thanks, Fackson. I have a question about the other group in the US that's working in similar area. Are you planning to collaborate with them since you know that they're doing similar research and just to avoid, repetition of work?
Yeah. So Tim Yoon, the group. So I met Professor Yoon, and we had a discussion. I met him in Dubai, and I was giving presentation, and he had some work on LinkedIn. They don't work on it anymore. And in terms of, because one thing that at the beginning we didn't know that they are doing it as well, and so it's nice to collaborate, as we said. But at the moment they are not working in that space anymore. But it was nice to be able to see that there are other people who have been working on LinkedIn and doing some nice stuff. But that was the most prominent group. And it was nice to be able to compare notes with them once I meet them at the conference. And we compared notes, and it was quite nice. Just a very share knowledge kind of situation.
Did that impact you being able to publish your work?
Well, it did impact in the sense that once you try to send, they say published. So you have to go to a lower, impact journal. Yes, you saw they published in JBC, which is quite high impact, and once it comes out and they got an award for that, so you can say, me too. I know the mechanism. So we did publish, but of course it was complementary to their work, and confirming we use different types of cells. And it was quite nice to see that they are seeing similar things. We did have a conversation about which residues, because they seem to be changing one, because they were more doing experiments where they change the residue, they remove the residue, and then they see if it works. They remove the residue and they see if it works. So we are not taking that approach, but it was nice to be able to try to see if you remove residues, it will still work. Yeah. So we have patented the short linkage, and they are, and they were really friendly and very nice to be able to collaborate with and to hear and what's going on, so that we also don't repeat stuff from here. Yeah.
Thank you so much. I just had one additional question.
Yeah.
About going back to etiology of the conditions that you were describing, and thinking immediately about sports and athletes. Do you see a lot of those conditions in high performing athletes?
Yeah. So one of the things is that for the, of course, we will direct a lot of this to more age order, but with the young people now, with all the athletes, there's all sorts of back pain, there's all sorts of early osteoarthritis, because whenever you have an injury to the joint, then what happens is that you follow the cascade of degeneration, the post-traumatic OA. And that's well documented that if you injure the joint, it's going to go into osteoarthritis. And so you start getting all these cases of young people with advanced OA. And but they are more likely to regenerate because their cells are young and chondrocytes, and they are more likely to be able to repair the tissue faster. But this is applicable to a lot of with all these kind of sports that will impact the joint, impact the spine. So that's the idea.
So now with what age are you diagnosed?
Well, I don't know. I think the generation, the older you become, I think maybe two. We have never done those studies, so I'll just speculate. But at least for the cells we are using, we are not trying to look at age. And it would be nice to look at age and also to look at sex in CHR and the NIH, where they want to see that you are doing and age together. Yeah, you see how active are the cells and how they are making matrix. That would be a really wonderful question to look at. At what stage can this technology not be applicable? Yeah, thank you very much. Thank you very much. Thank you very much.